So I'm happy to be here, and thank you to Jeremy to invite us to actually have this presentation. Just shortly about Apitope. Apitope has been as a company from 2002. It's a spin-off from Bristol University and the David Wraith Lab. David is still with us and acting as a CSO for our company. But in going back to autoimmune diseases as such, there is an autoimmune abnormality where the tissue or the immune cells is actually acting and combating their own tissue. And it can be life-threatening conditions like in Graves' disease, uveitis, MS, which I will allude to and report some data from. The ideal therapeutic, instead of having a general immune suppression, is actually to go in and reinstate the immune balance, because in all of these diseases there is an aberrant imbalance between T regulatory cells controlling the T effector cells, and the T effector cells goes off and be pathologic in this case. The T regulatory cells has been identified to either be too low in numbers or to have a function that is not sufficient enough to control the effector cells. The apitopes here, we hope, can treat the underlying cause of the disease and just leave the rest of the immune response to combat infections and cancer therapies. So what we use is actually synthetic, small, short peptides, and peptides derived from the autoantigen that is actually a part of the disease cause. And not all T-cell epitopes within an autoantigen is actually inducing tolerance, and that's where we have our platform to generate those data and be comfortable that we do use the correct epitopes. The peptides that we use are defined as apitopes. And with that we mean an antigen processing independent epitope. And I will come back a little bit about the mechanism behind that and why we call them. And they need to be able to be soluble and have affinity enough to induce, to reach the steady state dendritic cells in lymphoid organs and induce tolerance. To just show you a little bit about the property here and facing the solubility, this is a very short assay that we do when we have identified an peptide, which is about 15 to 20 amino acids long from the autoantigen. We can inject that subcutaneously to a mouse, wait 2 hours, isolate the APCs, the antigen-presenting cells, subsets, and then use an antigen-specific, peptide-specific T-cell line. And when we do a soluble peptide, you can see that the T-cell line is responding with gamma interferon production. But when you have an incomplete peptide with not soluble enough, there will be no response. So solubility is one key asset for apitopes and for tolerogenic peptides. Of course, the other requirement is affinity, because when they reach the lymphoid organ over here, they need to have affinity enough to bind to the MSC class 2 molecules. And here is where the ProImmune reveal assay is coming in very handy to provide us with that information. The target, as I said, is the MSC class 2 molecule on steady-state dendritic cells. And steady-state dendritic cells, they are sitting there canvassing and looking what's happening in the environment. So when these peptides are coming, and this is our hypothesis, they bind to the open MSC class 2 molecules. I say they are open, but they can have a very low affinity ligand bound to them. But nevertheless, when our epitopes are coming, they are binding to these open reading frames, and then open reading molecules, and then they can actually be presented to a T cell. But they will be presented in a special way. Normally, when you have a peptide presented by an antigen-presenting cell to a T cell, that will activate the antigen-presenting cell, and the antigen-presenting cell will express a range of co-stimulatory molecules, and the T cell will go off and be an effector cell with the sum of that signaling from the total co-stimulatory. And I said the apitopes are binding from the outside, which means that they just replace low affinity peptides and then bind to and interact with the T cell with the T cell receptor, the same one as over there, but without any range of co-stimulatory signals. This T cell will have an incomplete signal, which means that it will go into a regulatory phenotype. We previously called them monergic because we didn't really understand what they were called. So the key thing here is the peptide design, as I alluded to. Not all peptides will bind back into the MSC class II molecule in a proper fashion to be able to interact with this one when you make them as small, short peptides. So that is one of the keys, and where we have our proprietary platform to identify that we actually know that these peptides resemble the native conformation of the interaction. So interacting with the steady state dendritic cells will actually conform this naive, or could be a memory T cell, into what we believe is a TR1-like regulatory cell. And TR1-like is actually saying that they are induced in the periphery and not maybe coming from thymus-derived FOXP3 positive T cell, and I will come back to that. But this TR1 cell, through probably IL-10, but probably more factors that they assassinate, will have a feedback on the dendritic cell to actually downregulate the antigen-presenting function of this one. And that means that you will also have a bystander suppression on other T cells and other antigens that is expressed on the same dendritic cell. But it will also have an impact on naive B cells being matured into regulatory B cells, and that is important when you have an antibody-mediated autoimmune disorder, and I will come back to that. Showing you some preclinical data from our very first compound, that is ATX-MS1467, and we use a double transgenic model for this specific compound, which has a T cell receptor that is derived from a human for an MVP epitope bound on the DR2. So the OB1 here is the T cell receptor name, and the DR2 is an HLA transgenic mouse that we actually use. Because we derived these epitopes and we developed them for humans, that means that we used HLA transgenic mice. So if we do go back to ATX-MS1467, you can see that we do have a dose-dependent reduction when we give these epitopes, which is a cocktail of 4 peptides, into the mouse. And the treatment regime is most of the time twice a week or 3 times a week for all of those experiments. And you can see that even if we start just after immunization, but before clinical signs or in the beginning of clinical signs, we can really halt the disease and make it go back. But also when you start at the peak of the disease, you see that the high dose here is actually reverting the disease pathology pretty quickly, I would say. So I said mechanism of action here. So one of the first studies that was done was to actually look for cytokines. So this is done by EMD Serona under license for us. And what you can see in essence here is that after 7 days of injections, 3 injections of the epitopes, the cells was taken out and put in culture with the peptides as antigen. You can see that the proliferation here is actually downregulated in the peptide-treated mice, not in the controls. And IL-17 maybe not, but gamma interferon and IL-2 is significantly downregulated while IL-10 goes up. And I will come back to that because this is really the picture that we see constantly when we do these experiments, and is behind the hypothesis that we have an IL-10 mediated mechanism over here. To continue to say that if you start what's looking in the actual tissue, in this case it's spinal cord from these double transgenic mice after treatment, is that if you start the treatment day 7 with the first signs of symptoms or at the peak of the disease is compared to in the histology, and this is the control mice, and you can see how the structure is, or the disease is actually with hematox eosin is actually downregulated, and quite a lot if you start at the day 7, but it's still downregulated significantly and affected, even if you're protecting the tissue, even if you start at the peak of the disease. Demyelination in this case measured as Luxhal fast blue staining, and you can see that you have quite significant demyelination in the controls. If you start at day 7, you can almost prevent that from happening, but you still have a halt of the disease progress if you start at the peak of the disease, and the same is reflected in CD3 cell infiltration. So we think that the earlier you go in, the better response, but of course that's not really the case that you can do in humans, but you can still see a very good response preventing new things from happening if you go in at the peak of the disease. So looking at disease populations then and see what's happening. So this is 2-week treated mice, and then the cells was taken out. Here is the spleen cell ratio. If you have an HLA-DR2 binding peptide, or the peptide treated mice just analyzed by flow cytometry, and you can see that this is the LAG3 CD4 positive T cells are increased in the spleens of these mice. And also the FOXP3 negatives, IL-10 positive, is actually upregulated. Now you needed to have 3 days with PMI, ionomycin and monensin, of course, in culture. The difference in FOXP3 positive cells in these mice is not really significant or altered at all. So here is again an evidence saying that these cells in these mice is the TR1-like FOXP3 negative cells. Just to bridge over to the clinic where MRI is actually used as one of the major or primary endpoints. Here you can see an MRI study that was done in these mice to actually see if MRI was suitable to use as a biomarker, both for the safety and efficacy point of view. And the neurological disease scores is over here. And you can see that the ATX treatment in this study was actually started quite early, so you prevent anything from happening. But yes, the leakage volume is actually very different in this model, and the ATX is downregulated there. And again, the gadolinium enhanced intensity with the T1 weighted MRI, which is mostly used in the clinic, was also detectable as a difference in this case. So bridging over to the clinic then, because this has been in the clinic for 3 clinical trials, and I will come back to those ones. Mostly used as a first-line treatment in MS is still Copaxone. Copaxone is a randomly peptide cocktail that is actually injected subcutaneously to the patient. And here you can see that this big gray there is the Copaxone treated, and will benchmark ATX towards that one. And you can see that while Copaxone wasn't really active more to 50% or something like that in these models, starting at that time point you can revert the symptom, you can hold the symptoms with the ATX peptide treatment. Also with Gilenya, fingolimod, you can see that starting over there, just before first clinical sign, there is almost no disease in these mice, while the fingolimod here is the blue line, and you can see that then ATX is actually improving on the efficacy compared to fingolimod mechanism. So with that we went into the clinic, and this is the first phase 1A. and that is done quite a long time ago, and it was a very small study, 6 patients, but it's interesting because they are in secondary progressive MS. And to just mention about our clinical trial approach is that we inject intradermally 14 days apart. That has become our standard. I said in mouse we do every 2 or 3 doses a week, so we do it far apart. But 2 weeks apart is absolutely probably the best one in humans. We do a dose escalation, starting with low doses and up to the top dose, which has been for the clinical trial so far 800 micrograms intradermally. And the reason we do a dose escalation here is to slowly adapt the immune system to the peptide treatment and slowly start to induce the T regulatory cells. So going back to this data, it was really a very short study, so there is nothing to report about MRI, but what we could notice was that compared to baseline, 1 month after stopping the last treatment, you can still see that the MBP-specific T cells were lower had a reduced proliferation, but after 3 months, this was actually gone. And from a safety perspective, we analyzed the PPD response, and that was absolutely not altered, indicating that we just affect the T cells that is actually included in the disease pathology. The next clinical trial was the 1B, and now we went in with 2 cohorts, and this is 2 cohorts, and we did intradermal dosing and subcutaneous dosing. Again, the dose escalation up to 800, and then continue 800 for 4 times. So it was quite a long 16-week treatment period. And here is the MRI scores in active lesions from baseline. And you can see that following the baseline over to the first high dose was actually injected, there is no more difference, and it's high variability between the patients. But when you start to do the 800 dose treatment, you can actually synchronize these ones, reduce the number of active and new formed lesions down to 78%. This again seems to last for 4 weeks, but not for 3 months. Phase 2A, just shortly saying that the MRI data here more or less corroborates what we saw in the 1B, but taking into account that there was a shorter upregulation of the dose and a longer period on high dose, we think that after this study that going back to the slowly upregulation and prolong the top dose will be a successful regime to continue with. Interesting in this study was that the lesion volume was also evaluated. So it's not only that the new lesion formation and the active lesions were declining, it was also that the lesion volume in persisting lesions was actually also reduced. So that we thought was quite a lot of interesting, and going forward we will continue to evaluate that. One interesting observation, because this is a kind of few patients in this cohort, was that the MSFC score was evaluated as well. And MSFC score is actually a collated sum of 3 different arms: the upper limb function and the lower limb function, and then it is a cognition. So when we discovered that with these few patients, taking that into account, we almost had a significant reduction or change from baseline to end of treatment in these few patients. We splitted the different arms up and looked at them individually and could realize that there is a significant improvement in cognition using the paced auditory serial addition test called PASA from baseline to end of treatment. And of course this is absolutely interesting to continue into the next clinical trials to see what this can lead us. Safety profile. We have to date 68 MS patients that has been exposed to this compound. And as you can see, there is quite benign effects. There is some related adverse events, and they come from the injection site most likely. And the injection site has a transient reaction that resolves by itself after a number of hours, but that is from an intradermal injection. I would say that is probably anticipated. But there is no other severe adverse events that has emerged so far. And that is different from current treatments in MS patients, where you have monoclonal antibodies where they come with a risk of chronic infections and cancer development. Just a few words. I said we have antibody-mediated disease, and this is too much text, so just focus on this line. Anti-TSH thyroid-stimulating hormone receptor antibodies overactivate thyroid cells. They bind to the receptor and they trigger in an agonistic way, and they trigger the thyroid cells to overproduce free T3 and T4, and that results in an overproduction and a swelling of the thyroid. It leads in 50% of the patients to an orbitopathy, and there is currently no effective treatment. You can go on an antithyroid drug and that will wean off, and they have severe side effects, or radioactive iodine or surgery. So what we have done is actually in the same fashion as we did with the MS cocktail, to our platform produced a cocktail in 2 peptides this time called ATXGD59. And we have now been into the first-in-man study for this compound as well. So let's just go see what that happened. It was, the last patient out was in January, and we are now doing analysis of all the data that we have. But primary endpoints was of course safety, and it seems to have the same safety and tolerance profile as the MS drug has. And then we have all the other data with free T3, T4s, and the antibodies, stimulatory, inhibitory, and also total antibody levels that we are now looking into for the full cohort. But we did an interim analysis with the first 5 patients that was included in the protocol that we needed to do. We do first-in-man studies in patients already, not in healthy volunteers. And that is because when we went into the clinic with the first phase 1A, for the MS, that was a recommendation from regulatory, because what they didn't want to do is actually have the risk to induce disease in naive patients, but with having a potential to actually develop the disease. So we stick to that, and regulatory has approved that so far. And to say that the safety profile in these patients now for the first 5, we have still a number of 5 more to analyze, is looking promising, and we have initial signs of efficacy when it comes to T3 and T4 return to normal limits in the majority of these 5 patients. We need to now see that holds up for the next coming 5 as well. And we also noticed a reduction in the anti-TSHR antibodies in those patients, correlating with the free T3 and T4. But take this data as very interim, but it's looking promising, and it's looking like we can downregulate an antibody-mediated disease in the same fashion as with the T-cell, more T-cell-dependent disease, directly T-cell-dependent disease like MS. So my last slide is actually just to give you that we have the platform. We can develop the apitopes that has the potential to downregulate and induce tolerance and restore the tolerance and the balance with T regulatory cells, which we believe is the TR1-like cell, at least in mouse. We need now to prove that in humans. We have safety and efficacy data from 3 phase 1s and one phase 2A study, 78 patients in total, and it looks very nice. It's safe and well tolerated. So our prospect now is that within 12 months we can have 3 phase 2 products in going further to the phase 2b in multiple sclerosis with the MS compound. And with the GD compound, we are preparing for the phase 2, expecting to start in end of this year, and full data set will be available in Q2 this year for the phase 1. But from what we have seen, we plan for the phase 2. And then we are going in an orphan intermediate uveitis with a third drug, and that will also happen probably this year. So in conclusion, we do have for 1467 is halting disease progression, and we see that in the animal models, and we probably see that in the patients with the new formation of lesions halted in patients that is actually treated with the MS drug. It has excellent safety as well as efficacy. We have the cognitive function that we need to go further with and understand, and from animal models it's also suggesting that the remyelination can happen, and that is now also supported by recent publications saying that Treg regulatory cells can drive the maturation and differentiations of precursor cells to oligodendrocytes. And I think that was my last slide. So thank you very much for listening.